Hello everyone, my name is Simona Itri. I am a chemical engineer and a second-year PhD student in mathematics, physics and application for engineering at the University Vanvitelli in southern Italy. I am doing my research activities at Simonetta Grillis lab in the Institute of Applied Sciences and Intelligent Systems of the National Research Council of Italy in Naples. Specifically, I am working in the framework of a European project called SensApp, which aims to develop an highly sensitive sensor for detecting Alzheimer's disease biomarkers. And now let's jump to my presentation. Today, I am going to talk about one of our recent works, where for the first time we used a new technology for the detection of extremely diluted organic molecules. This technology is called Feeroelectric effect, and this research was a collaboration between our lab and the Technical Research Center VTT in Finland. So, why is it so important to detect extremely diluted organic molecules? Because some key biomarkers may present at very low concentration in blood samples. Therefore, improving our ability to reliably detect and quantify organic molecules means improving disease diagnostic tools. So, in this work, we tried to overcome some of the technical challenges related to the detection of substances at very low concentrations. However, we were specifically interested in the detection of biomarkers, which are molecules, typical proteins, used as probes to diagnose diseases. In an ideal scenario, we would work with a disease-specific biomarker. We would be able to reliably quantify it and detect it at the early stages of disease, so that if treatment is needed, it can be timed appropriately. However, in reality, disease detection is a complex process where most typically biomarkers at disease onset are present at extremely low concentrations in bodily fluids, and this may hinder our ability to correctly diagnose a disease when treatment could be most effective. Although nowadays a diagnosis of the Alzheimer's disease already exists, it is very invasive and requires the hospitalization of the patient. In fact, this diagnosis is carried out by analyzing the cerebrospinal fluid obtained by a lumbar puncture. There is currently no way to diagnose Alzheimer's disease by using blood samples, which would be a far easier and less invasive method. Indeed, as I said, blood analysis is less invasive and far more cost-effective. In the case of the Alzheimer's disease diagnostic process, the most common technique is the ELISA, done on cerebrospinal fluid. The ELISA is very widely used and highly reliable. However, this technology has the downside, which has a low limit of detection of biomarkers, which is in the range of 50-100 picograms ml. This concentration is too high compared to the concentration of biomarkers in blood. In this scenario, the research project SensApp, which is the framework of my PhD, is trying to push the limit of detection of Alzheimer's disease biomarkers to way lower levels. SensApp stands for super-sensitive detection of Alzheimer's disease biomarkers in plasma by an innovative droplet split and stack approach. I'd like to mention the partners of the SensApp project, which are VUB University of Brussels, IRCS Centro Neurolesi di Messina, the University of Linz, the Technical Research Center of Finland, and Genovis. Regarding the technical aspects, in our research activities, we use the pyroelectric effect, which is a property of a certain crystal, which are naturally electrically polarized. Pyroelectricity can be described as the ability of certain materials to generate a temporary electric field when they are heated or cooled. A pyroelectric material, like the lithium niobate crystal, at room temperature has surface charges in equilibrium with the external charges. As shown in this figure, the thermal simulation of this crystal creates a condition of unbalanced charges, which creates an electric field. Here, we use this electric field to dynamically manipulate fluids. Specifically, we used a new technology called pyroelectrohydrodynamic jet, where using pyroelectricity, we create an electric field which modifies the meniscus of a very small liquid droplet. As shown in this figure, we use a crystal of lithium niobate, which is a pyroelectric material, placed over a microscope glass cover slide. When heated, the lithium niobate generates an electric field, as a consequence of which a liquid droplet placed underneath the cover slides changes in shape. Specifically, its meniscus evolves into a conical shape under the action of electric, capillary, and viscous forces. As the electric potential increases, the droplet changes in shape into a conical profile called Taylor cone. This Taylor conic profile is maintained until we reach a specific threshold of electric potential. Once this critical limit is reached, the meniscus profile can no longer be maintained by the surface tension, and jetting initiates from the Taylor cone tip, which means that a number of very tiny droplets are produced, as shown in the black and white snapshot on the right. This process was developed by the Institute of Applied Sciences and Intelligent Systems and has been called the pyroelectrohydrodynamic effect, or more simply, PJET. So why is it so advantageous to induce this jetting process in a liquid droplet? Well, one of the main applications is the bioaccumulation of some molecules. And here I show the process of bioaccumulation in a new vertically flipped configuration of the PJET setup developed with the contribution of the University of Brussels, with an approach that we defined split and stack. As shown in this figure, we first load a droplet of fluid onto the loading plate. This fluid contains the biomarkers. The droplet is forced to pass through a micro-orifice, and under the influence of the pyroelectrohydrodynamic field, it's forced into microdroplet as shown in the step 2 and 3 of the figure. As a consequence of the stacking of these droplets in a very confined area, the biomarkers are layered one on top of each other and bioaccumulate. Thus maximizes the chances of them interacting with an antibody, therefore resulting in the formation of an immunocomplex product. Facilitating the formation of such immunocomplex when the original concentration of biomarkers is very low is the key to disease diagnosis. Another advantage of our approach is that it requires very low volume, around 20 microliters, hence which is the key to working with the rare, expensive biomolecules. Here we test the accumulation ability of the PJET system using a fluorescent dye, the Alexa Fluor 647, and a confocal fluorescence scanner comparatively to a commercial printer called Dymatix. This work was done together with VTT, who tested the Dymatix while the PJET was tested by our group in Naples. This slide shows the design used to test the PJET using the fluorescence dye Alexa Fluor. To test the hypothesis that increased deposition onto a confined spot would drive bioaccumulation, we quantified the fluorescence signal emission using the fluorescence scanner InnoScan. Tiny droplets were pyroelectrodynamically released onto a confined spot of 100 micron in diameter through the process I explained in the previous slides. Using a fully factorial design, we test the 3 increasing concentrations and a PBS solution without dye used as a control. We also test the 5 spots types produced with 1 to 5 droplets. Across tests, we analyzed a total of 160 samples. The Dematic is a piezoelectric inkjet printer which has been developed for laboratory process development and materials evaluation. It has several applications, including fluid and substrate interaction evaluation and deposition of biological fluids. The tests using the Dematics were carried out using the same design explained before, with the difference of a lower number of replicates and a different buffer, which were all design choices driven by technical consideration of most efficient use of the system. This test quantified the fluorescence using a scanner with the same technology as the InnoScan used in our experiment. As shown in the figure, we demonstrated that the increase in droplet deposition drives an increase of the fluorescent signal with a linear relationship. Therefore, our initial hypothesis was confirmed. The same result was demonstrated across the 3 tested concentrations. Here is the output of the fluorescent scanner. The black dots are successfully released droplet quantified by a confocal scanner. However, as we can see from the image, there were missing spots indicated that dispensing might have been unsuccessful. Here are the results collected using the Dematics. The droplet sizes were comparable to the PJET technology. However, sample spots have random fluorescence intensities. So we did not find the same evidence of a linear relationship between droplet deposition and fluorescence using the Dematic technology. Considering the successful results, we decided to test the PJET technology using lower, more challenging dilutions. Therefore, using the same design as before, we test the 2 additional concentration of 1 picograms ml and 0.1 picograms ml. As shown in the 2 figures, we proved the same linear relationship between droplet deposition and signal intensity. I believe it is really interesting that we found evidence of a stronger linear relationship, as shown by the higher gain, with the more diluted samples, suggesting that the PJET system may be most efficient in the bioaccumulation of very diluted samples. Even more interestingly, we were really able to push the limit of the detection of the PJET system to extremely diluted biomolecules. Indeed, the lower test concentration was 0.1 picograms ml. Now, I want to say many thanks for your attention. I hope you have appreciated my research work. Very special thanks to Inka Pakila from BTT team and the European project SensApp for founding this research. I wish you a very nice day and enjoy your conference. Bye.